morning, everyone. Thanks for coming to this webinar. I'll be talking about some of the genomic analyses that we've been doing on some of these elms that have been talked about already. And as has been mentioned, this is in collaboration with Joan Webber. And Mohammed Vatanparast is the postdoc who's been working on this project. All of the analyses that I'm going to show you today were done by him. And we've also had the privilege of working with Juan Martin at the UPM Madrid, who supplied a lot of samples, including lots of hybrids that they have in the breeding program in Spain. And you'll see more about them shortly. So we took a whole genome sequencing approach, and we did this on 180 samples of elm. And we did them at 20x coverage. That meant that across the whole genome, we could identify with some confidence the single nucleotide variants. So this could not have been done before because the technological advances simply weren't there. It would have been prohibitively expensive to do this before now. And also, we've only recently, as a scientific community, gained chromosome level reference assemblies for elm that we've been able to use. And most of what I'm going to show you today is a principal component analysis to summarize the variation in the samples. This is a method to try and boil down all of the variation across the whole genome as much as possible into two-dimensional figures. So here is the first and second principal coordinates of our data. You can see this kind of triangle shape, and what we're finding is Ulmus minor is over here at one corner, Ulmus glabra at another corner, and Ulmus pumila in another corner. I'm not showing you Ulmus americana or Ulmus laevis in this picture because they are quite different to the others, and that if I included them, what you're seeing here, which is probably of most interest to us today, would all be kind of a bit scrunched up. You can also see various Asiatic species are clustered in the middle. Now, that's not because their variation is intermediate between these, and we can see that when we look at principal coordinates 3 and 4, those pull out the Asiatic species. So the Asiatic species are quite different. It's just that because there are not many of them in the main PC1 and PC2, they're not coming out as very different. Reassuringly, we find that known hybrids are coming out where we would expect them to come out. So, for example, Fiorente is known to be a hybrid between pumila and minor, and it's coming out in between the two. Similarly for Den Haag, Patriot, which has 4 different species within it, is coming out sort of where we would expect it to. Wingham, which has minor, pumila, glabra, and wallaceiana inside it, comes out kind of intermediate. Sapporo Autumn Gold and New Horizon, which are both known to be hybrids between Davidiana and pumila, are coming out between those species. So it all seems to make sense. Here's a family tree for a cross that you're going to hear more about in the next presentation by Kaz Gudgeon. It was a cross done by David Hurling between Wingham and Ulmus minor at Tong Mill, and you can see the different parents of the cross, and some open pollinations also occurred in the origin of Wingham. Well, if we look on our PCA, we can see the parents here. We had a glabra component, a pumila component, a wallaceiana component, and then this is one of the parents of Wingham, FL493. 1038 was open pollinated to produce Wingham, and it's interesting that Wingham has moved slightly towards the middle, not simply towards Ulmus minor. So we suspect that the other parent of Wingham was also a complex hybrid. And this is where Tong Mill falls, which is the British elm that David Hurling crossed with 493. You'll hear a lot more about that from Kaz in a moment. I want to focus now on this line that runs between Ulmus minor and Ulmus glabra. And I'm going to use the term Ulmus hollandica in a very broad sense for things that are hybrids between the two. Here's something called an admixture analysis of all of those trees that run into that line. So some of them are pure minor, they're just orange. Some of them are pure glabra, and then you can see some of them have admixture between the two. Some of them just with a very small component of glabra. And that maps directly onto this line that we can see in the PCA. So up here we have pure ulmus minor. Some here have sort of less than 10% ulmus minor towards this end. Then we have admixed individuals, and then we have pure glabra. And here we are just sort of zooming in a little bit on those individuals that we've included in this admixture analysis. So you can see a range of names in there, some of which many of you may recognize and that we've heard about before in this talk, these talks. I'll just look very closely at the cluster that includes the ulmus minor end of this spectrum. At the very far end, we have a cluster that comprises some ulmus procera from Matthew Ellis's collection, 4 ulmus minor individuals from Galicia, and 2 ulmus minor from Brighton. We think that this is probably the Atinia clone, the ulmus procera in the strict sense, which is thought to have come from Galicia to the UK, brought to us by the Romans, of course. We then have separated from that a cluster of ulmus minor in the broader sense. Then towards the edge, we have some individuals with a little bit of glabra, and then down here we have individuals that have much more glabra within them. And some of them are called Ulmus hollandica, which is what we would expect them to be called. Now, Sell and Murrell named 63 different species of UK elm, and we had not all of them, but we had some of them in our sample. 11 of them were supplied that had been verified by the experts, Brian Evesham and Alex Prendergast. And we can see that most of them are falling into this broad cluster of Ulmus minor, and 2 of them, as expected, fall into Ulmus glabra. Sell and Murrell said that they were, closely related to Ulmus glabra. So what these species names seem to be telling us is they're useful labels for certain forms of Ulmus minor, but I think it's clear they shouldn't be viewed as species in the same way as Ulmus minor and Ulmus glabra and Ulmus laevis, etc., are viewed as species. And if we look very closely at the cluster where the Sell and Murrell species are falling, we can also see that they're falling with other individuals from the UK and indeed from France that have been labelled as Ulmus minor. Also note that Reddii and Goodyearii fall very close to this cluster as well. And here are the 2 Sell and Murrell species that fall near glabra. They are slightly differentiated, but within the realm of what we would call glabra. Now, 7 Ulmus minor clones were released from Spain as selections of Ulmus minor from the wider environment that have survived Dutch elm disease. They were released a few years ago. The most famous of them is Adamers. In our results, Adamers seems to be 27% Ulmus glabra in its genome. It falls well within the range of individuals in the PCA that are integressed with genetic variation from Ulmus glabra. The same is true of Dehese de Amaniel. It's 18% glabra. And then Retiro and Dehese de la Villa are both, we think, pure minor. But it's these 2 that are the ones that are most resistant to Dutch elm disease. So throughout the talks, we've tended to call Adamers Ulmus minor. Actually, it might not be Ulmus minor. We're still checking this result. It was a surprising result, but at the moment, that's what our data seem to be saying. There's also some new light on other accessions. So Ulmus minor Amsterdam and Ulmus minor Beer Schwarz both seem to also have considerable amounts of glabra in them, and so perhaps should be seen as Hollandica rather than minor. We had a collection from Frank Matthews that was called Dodwell, Ulmus procera Dodwell normal leaves, and one from Staffordshire. Those don't seem to be Actinia, they seem to be minor introgressed with glabra. We also looked at the range of individuals between Ulmus pumila and Ulmus minor, and again we found evidence of introgression of pumila into minor. And interestingly, some of these were things that have been identified before as minor or even as Hollandica. So perhaps there is a component of pumila in them. We also looked at the genome-wide pattern of genetic variation in Ulmus minor and Ulmus glabra, and very noticeable that Ulmus minor contains considerably more variation. You'll see the scale for Ulmus minor goes from 0 to 2,580 SNPs per megabase, whereas in Ulmus glabra it's only up to 522. So we're finding a lot more variation within Ulmus minor than within Ulmus glabra, and that's in agreement with a previous study by Ewan Bowditch, I believe. So here's a hypothesis about what's going on with UK elms. I think we have a little cluster that is Actinia. Then just below a Spanish cluster, we have what is perhaps a native British cluster of Ulmus minor in the broad sense, including many of the Sella morel species. Then the edges of this have variation due partly to introgression from pumila and partly to introgression from glabra. And then we have a whole range of elms that contain more glabra. So in conclusion, admixture between minor and glabra may have been more widespread than we imagined. Ulmus minor seems to be more diverse than glabra in Britain at the genetic level. We've placed the Sella morel species firmly in minor, or some of them firmly in the minor cluster. We've shed new light on the origins of some widely planted clones, including Adamers. Some living collections contain mislabeled specimens. I haven't done a slide of that, but we did uncover many cases of that. And in conclusion, DNA variation within elms in Britain is clearly higher today than it's ever been in the past due to all of these introductions. So there's lots to be encouraged about for the future of elm in Britain. I'd like to thank everyone who supplied samples. It was a large number of people. And I'll just remind you, Mohammed Vatanparast is the postdoc who did all of the analyses